Welcome to my presentation, in which I would like to give you an introduction, a beginner's guide into how we do data analysis in the case with transcriptomic sequencing data and how we can use Snakemake workflows and R in a fairly reproducible manner. Just to start, this presentation is for a diverse audience. That means everyone who's interested in data analysis, irrespective if you come from the vet lab, you're a biologist, you know everything about how to make the experimental chemical side of sequencing. You're just curious how data analysis might work. Or if we, let's say, you come from the computational side and you've never worked with sequencing data and you're just interested, what does it feel like to work with data analysis in the sequencing world, this presentation is for you. And now to just to have the heads up what I would like to present here. I would like to, of course, for those who have never had the experience to work with sequencing data, I have to give an introduction. What is it like to work with it? What is transcriptomics even all about? Give you an introduction to what the overall structure of sequencing data is, how we might be able to analyze it if we go for a simple bash pipeline, for instance, and what kind of downside it might bring to us, and how Snakemake might address such of these advantages, or how I like to call it, how I'm trying to avoid using bash as much as possible. And then finally going into how can we use the data that we produce in a workflow that also involves R scripts, how to do that in a more reproducible manner, and get also a bit into what does it mean to be reproducible, how to take care of software installations, how do we manage that across different systems we might work with, our workstation, our notebook, maybe a nice computer cluster, how could that maybe be involved, and of course finally I hope I can give you a bit of fun introduction to various R packages and little tips and tricks that you might like to have. In case you're interested, the slides, they are on my GitHub page, and the tutorial that I will refer to later is also there on the links that I've shown here. Now, just to begin with, if we think about data analysis and big data, you might like to think about these huge, big, astronomically large data sets, but reality is, the life science, we've outgrown astronomy. So we're not just doing astronomically large, we're doing a life science large data sets. And if you look at numbers, for instance, here from this paper from 2015, we have a dozen times more exon bytes of storage needs in the genomic area of life science for just to handle storing all the data. But it's also a bit here to highlight the difference. For instance, to compare it to astronomy, we have very heterogeneous data. So depending on what type of study we're looking at, maybe you have a population style, large disease genomic association study, and you have, I don't know how many terabytes, maybe a petabyte of data, very somewhat more homogeneously designed. But if we then say we are more into elementary research of various samples, as soon as some sort of genomic structural analysis is involved, the data set becomes much more heterogeneous. We might have to have a much more hands-on approach to process the data, which is maybe in contrast to astronomy, where there might be one large satellite dish producing I don't know how many terabytes of data per second, and it's just about reducing the data as quickly as possible to be handling an analysis workflow. So here, genomics, we might have quite diverse needs for the analysis. And in case just to recap, if your biology high school lecture has been a while ago for you, if we think about a genome, that is this double helix DNA structure. You might recall it was with these 4 letters of nucleotides, T, C, G, A, that make up the blueprint of life. And there is an RNA polymerase that can copy a certain part of the genome into an RNA transcript, which then can be translated to a protein, which after successful folding will carry out the function in a cell, or at least that is the traditional view of the central dogma of molecular biology, if you heard of it. Let's assume you're an architect or an engineer with a blueprint of a project you're working on, and you would like to have help working on it because it's not humanly possible to build a car all by yourself or a building. You need construction workers. You have some suppliers that provide you with handy tools and all the nuts and bolts that you might need. And let's say there's a very specific screw that's not standardized that you need for your project. And you go to a manufacturer and to request that they produce this screw for you. You don't go to them and give them your whole blueprint. It's just too much information. They might not really know now what you want. They will be overwhelmed. There may be also information on there that's proprietary. You don't want to give away. So what do? You make a copy. You make a copy of that specific part of the blueprint that is needed for the manufacturer to provide you with the screws. And if you now think about this word of transcriptomics I've mentioned, it is the suffix "-ome" you look at the totality of some sorts. In transcriptomics, we look into what type of transcripts are there in the biological cell, so we get an idea of what is going on. Similar to if we have now a complex engineering project and something might go wrong, you have to maybe take now a step back and look into what have I copied, what have we ordered, have we maybe missed a part, to have an idea how the system of your project comes together. And the same here in biology. We would like to know what is going on in a cell, because differences in expression is what drives all the differences. If you think about, let's say my neural cell in my brain and my skin cell, they have both hopefully the same genome, but they will do completely different things in life, the so-called phenotype that we see. And that is due to differences in cell expression in the development. And if we would like to know what's going on in expression, of course, we have to keep in mind what type of cell tissue are we looking at, what is the genome, what do we know about an environment or a certain exposure to drugs or radiation that could influence all kinds of expression. And of course, the for pharmaceutical research interesting part of looking into potential association to disease. And can we maybe determine what brought about it, undesired change in expression, how to maybe fix it to help people. So that is in short what transcriptomics is about. We want to know what's going on in a biological cell. And how would we even look into a cell? If you think about the illustration of a radio, you want to know what's going on in a radio. You maybe go into a tool shack that you have, pick up a screwdriver, and do some screws, open up, get an idea what's going on in it. But there is no standardized Phillips screw to open up a biological cell. So we need a certain other way to open up the cell and to sequence what has been going on. And that is literally the term or the tool the biologist used. It's a shotgun that blasts the cell into small pieces, use a chemical assay to extract just the DNA or RNA, prepare it in a way that it can then be placed in a sequencing machine. And the sequencing machine gives out a lot of small sequences that, if we were to simplify it, our bioinformatics job is to put all those pieces back together and get an idea what might have been going on in the cell. And in case you wonder, yes, that is the technical correct term to say that it's shotgun sequencing, to blast everything apart. And if we analyze such sequencing data, I would divide it into three main parts. One is what I would say the pre-R or Python analysis bottleneck. We provided with a lot of sequencing information, which is maybe terabytes of dozens, hundreds of essentially compressed text files, which we have need for specialized tools to work with. So we have a lot of data, a lot of long running times. So that's like this top of this cent-hour class that we have to spend to bring it into an intermediate result, which is just a count matrix, which is, let's say, per patient, per column, or per sample that we have, per row, a certain gene, a number reflecting how highly a gene is expressed in a patient or in a sample that we want to look into. And only then we come to the data explorative part on the bottom of the cent-hour class, where we can now spend, of course, a lot of time again to analyze what's actually going on biologically in it, where the statistics come in. To get an idea what sequencing data might look like, I would point to this paper from Jaime et al. From 2014, in which they've studied the influence of various estimate drugs on the expression on muscle cells on cell lines. For those who maybe are not as aware in the biological part, cell lines being what has been grown in a petri dish, so on a lab environment, not from a patient, which gives us the opportunity to even work with this data set because it's freely available. There's no ethical concern working with it. And if we would like to work with this data set, we need, first of all, our human genome sequence. I would suggest to go, for instance, to the GenCode website, click on the website around until it gives us a download button where you can copy paste the link to a URL, which we can then download from. And now comes the first bash script that is here indicated on the right with these dollar signs. That is, I type a command, for instance here, wget webget, to download a certain file from the internet and store it in, for instance, genome.fna.gz. In case you're not as familiar with bash, that is a way how to interact with your computer in a very programmatic way. Maybe most people might be familiar with these old times of Windows DOS, where there was no graphical interface, only typing. And we still have these kind of terminals in computers, on servers. It might take a while to get used to, but it's a very efficient way to interact and with a computer and tell what we want to do. And for instance, in this case, I make myself a variable URL and then tell wget below this folder on the web page, download the genome assembly and the corresponding annotation as an FNA and as a GFF file. If we now look at the files with, in this case, I would suggest to use the gunzip command to look into this compressed genome file as indicated by the gz suffix, to then have a look and see first line telling me with a greater than symbol that the chromosome 1 sequence is coming, which first starts with a lot of ends, meaning it's not a specified nucleotide at the very end of the chromosome due to the telomerases that protect our chromosomes from aging. So it's not as well defined how exactly the sequence there might be from these repeats from the sequencing. But once we skip a couple of hundred lines, then we see finally these nucleotides that we are familiar with, T, C, G, and A. And here finally we see, well, the human genome sequence for chromosome 1. And if we skip yet another few thousand rows, at some point we come again to a line that starts with this greater than symbol, which tells us now comes a new sequence, in this case the chromosome 2. And then it goes on and on until all the human chromosomes are described. This file, when compressed, is a bit under a gigabyte large, but if we actually uncompress it, it's 3 gigabytes roughly, so it's just a pretty large text file. Having the sequence alone is of course nice, but we also would like to know the annotation of genes that is in this GFF file. So if we look into that in a similar manner, we see that at first with the pound symbol indicates comments that tells us here comes a GFF formatted file, a generic feature file format, and the description is we have an evidence-based annotation of the human genome, and of course as scientists we like any kind of information that's evidence-based. And finally then the annotations themselves is in a tabular format telling us on which chromosome are the genes located. For instance here we know that on chromosome 1 between 11,800 to 14,409 is a gene, which in the last column it tells us is named DDX11L2. Since it might be a bit tedious to look into gene annotations only in this text format, we humans like more visual representations, for instance as a genome browser. So on the genome browser we can look into the annotations and think about the genome now as a one long string in which different colored highlights indicate where genes are. And if we zoom in, we see, as shown here, the sequence and the range in which the gene is located. Lastly, I would like to point out this +1 indication from our annotation file. That means, or tells us, is the gene located on the forward or the reverse strand, meaning or if you remember the figure of the DNA, it's a double helix, so there are two molecules, two strands, and they are reverse complementary, and depending on how the transcription and translation is, does it refer to information that was on the forward or the reverse strand that is encoded with this plus and minus in this column. Now, lastly, now that we have the genome, we would like to know the transcriptomic data that I've pointed out. In the paper from Himes et al., it at some point states the RNA-seq data is available at, then it states the name of a database and some more or less cryptic sequence of identifier that denotes the entry in the database. And if we use Google or DuckDuck to search for it, we quickly come to the sequence read archive at the National Library of Medicine in the US, and it contains the transcriptome analysis for this asthma airway muscle-related study. If we click around again on this website, we can either directly download it from there or even use a specialized tool, the SRA toolkit, to help us with downloading it, which is helpful if we have a couple dozens or hundreds of files that we would like to download. In our case, let's say we just downloaded a handful of datasets from the website, and I have now on my computer the genome annotation and the sequence that we just downloaded earlier, and in a folder I'll call raw data, we have multiple FASTQ files. These files are named in that way, that SRR, meaning sequencing read run, so it's the identifier from this database, and the sample 1039508 refers to a control sample, and 509 to a asthma drug treated case that we want to look at. Each of these files are more or less about a gigabyte large, but once uncompressing them, they can be up to a dozen gigabytes large files each. And you might now see, okay, let's look into these files, and might have noticed here's a _1 and a _2 for the same sample name. So what is that all about? If we think about a human being as a picture, we might have a front, a face, and our back. But if you have these two pictures from the same person, that's still the same person. And similar in cases of sequencing, after our lab technician or biologist made a shotgun sequencing, we have these little pieces of RNA, and they were sequenced maybe with paired ends, meaning it was sequenced from one and from the left and the right, or in biological terms from the 5' and 3' end. So there's two sequences that belong to the same molecule, to the same fragments. And these files contain, here with _1 indicating all the reads from the front, and _2 essentially all the sequencing information from the back. And if we now actually look into such a file, again using gunzip to look into this compressed file, we are again confronted with a long text file which has the following structure. First, we have this @ symbol indicating now comes a sequencing entry with SR R1039548 being the sample name again from the file, and .1 meaning this is the first read. The rest of the line is encoding details such as what type of sequencing was used, what was actually the serial number of the microscope cell or the flow cell that was used, with details on where on this sequencing device the sequencing happened. But the interesting part is here we see CATTGC. Yes, those are those letters we know. That is our genome sequence, and it's a small subset from the genome that was sequenced, and we would like to know where it comes from. Then it has this plus sign, repeats whatever was the text on the @ 2 rows above, and some more or less cryptic name that will come out in a moment with. To illustrate what our goal is, we have this short sequence from a sequencing experiment, and we would like to know where in the genome did it come from. So visually, if we think about the genome browser, our long genome, we'll zoom in and we would like to highlight where did we see evidence of sequencing, where did we have a read from, and if we look at the bigger picture of all the reads, where they are, we want to know, does that overlap with any kind of gene? So in this case, we have the KLF15 gene, and we see here a set of different read pairs indicated with the front and the back that I mentioned of this paired end sequencing, and the pairs connected with this line. And we would like to know how many read pairs map within the range of our gene, and that is our number representing the expression value of this gene in our sample. How would we get there? I mean, we have our genome sequence, we have our RNA sequencing data in this FASTQ file. All we need to do is now just look up the sequence, right? And then we check for the overlap, have our gene expression value. That should be straightforward. Well, it's a little bit more complex. So there was this fourth line in the FASTQ file with these cryptic characters. They encode the probability that a sequencing error occurred. So the sequencing machines, they themselves check the signals while sequencing if how reliable the nucleotide was determined. And depending on what type of manufacturer for the sequencing machine we used, they use a printable interval from the ASCII code characters to represent the -10 times log of these probabilities to determine if that is a trustworthy nucleotide that was sequenced or not. So we have to first process the raw sequencing data to get rid of potential wrongly sequenced or highly probable erroneous sequencing reads. Another aspect is efficiency. We have short reads here, just a bit under 100 nucleotides or characters if you think of it as a computer scientist. Per file, per sample, we have, in our case of this data set, somewhere between 13 and 34 million pairs of reads. And the human genome is more than 3 billion base pairs long. So we have a lot of reads, a large genome. We can't just do a simple reading the genome front to back and check if the read came from a certain region. We need a more efficient algorithm. And luckily, such algorithms are available. For instance, we could have Boris-Wheeler style transformation of the genome, which gives us an index to look up where a read might come from. Think about it like a phone book. You might remember the good old times of having paper-based phone books. You had the name of a person you want to call. You quite efficiently can look up the name and see, Miller Thomas lives in which city, which address, and a phone number. And similarly, it's with sequencing data. We build an index of the genome. We look up for a small name for a small sequenced read, where in the genome it came from, and then from there build up our alignments or mapping of these reads. An alternative would be to use approximation, for instance with a sequencing spectrum. Think about it like a light spectrum. If you have the wavelengths of light and the different abundance or strength of intensity. If I look at my light bulb on the ceiling, it has a somewhat yellowish color. And if I look outside to a beautiful Danish gray cloudy weather, that's more gray blue. And if I now find from my screen that I look in front a very bright color, I might be able to associate with which type of lamp might produce such a picture. And in case of sequencing data, we look at the k-mers. For instance, how often does with k equals 5, the A, so 5 times A appear in a sequence. And we go through all 4 letters, so ACTG to the power 5, and get then an idea to approximate where in the genome a read came from. The general outline of when we have sequencing data and want to process it is as follows. First, we have some quality control step on the sequencing reads. We build for our genome of interest an index. We combine those two to make a gene mapping. So we now know where in a genome a read came from. And together with the gene annotation, we can then check for overlaps and count the number of occurrences of reads per gene to make this gene expression matrix, where then finally we can come into our subsequent data analysis. So how could we do that? That comes now back to this idea of having a terminal, like a bash shell in front of you. We could type what we want to do. Let's say I have a hypothetical quality control tool called QC tool. I could, for instance, simply type QC tool. Please work on raw data for sample number 1 and place the output in, let's say, the folder clean data under sample 1. And we repeat that for every single step. If we have a lot of samples, that might be tedious to type and copy paste for all our samples. Instead, a slightly better solution would be to make a loop. That is, for instance, we could type for each FASTQ file and raw data, I have a variable I extract the file name and say QC tool, please work on my raw file and store it under clean data. Similarly, we could now extend and say, okay, we have one for loop that works on each sample for the raw data, provides the clean data. We have a hypothetical tool to build an index, have a second loop that for each clean data file uses a hypothetical mapping tool that takes the genome index and the clean data to produce some mapping file, which then can be used together with the annotation to count the number of gene expression. In case now to point you to the tutorial on my GitHub page, I have a small tutorial where I made this hypothetical concept pipeline that works just on empty files to see if we run it. It tells me each step it would be running. So in our case, if we start with some samples in our raw data and the genomes sequences, it would run indeed the QC steps, the index, and the mapping tool per sample, which gives us at the end then a folder with clean data with a sequencing data processed per each sample, the mapping file against the genome, and finally the matrix that we can then use to use our downstream analysis. However, here now we might want to take a step back and look at, okay, we have now the concept of how this pipeline might work, but there are quite some downsides to it. What happens if we now get a new file, a new sample? How do we rerun the pipeline? Would we overwrite all the files we've had so far? The for loops would not allow for that. And given that all these cleaning and mapping files take a considerable amount of time, we don't necessarily want to redo work and overwrite existing files. Also in this for loop, if the samples are independent, we should be able to run that in parallel, either on the same machine or even in a cluster on a completely different node. With this bash script, it's not that easy to do. How do we know if we have consistency? Let's say we would like to change a parameter, use maybe a different tool of quality control. How would we be able to run it and know that the mapping or the count matrix at the end is consistent with what we had in the beginning? And how do we keep track of software installations? Now, of course, in this concept pipeline, I used hypothetical commands, but how would we know how to install them? Where do they come from? If I come back a year or two later, and the software version might have changed or it's no longer installed on my machine, how would I be able to run the pipeline again and check how the result turned out? Now that is where Snakemake comes in. Thinking about Snakemake, it's a workflow management system that gives us the ability to run analyses in a fairly scalable manner. So if we have a data set with a lot of samples, so that is the x-axis, we should be able to quickly run steps that are independent from each other in parallel. We want to automate it in a way that if, let's say, we have a sample with this red processing indicating step, all subsequent steps should be automatically initiated as soon as the preceding step has been done. So there's a scheduling involved, but also the intelligent consistency checker. What is happening if, let's say, the red step had a slight change in parameter? Snakemake is smart enough to know, okay, I also have now to rerun all the steps because the data might have changed. And of course, the nice part with Snakemake, it's also portable, meaning I can run it on my notebook, I can run it on a computer cluster, on cloud computing. I can handle all of these parts. And the way I like to think about Snakemake is as a cook. I have a cook and tell them, please prepare for me. In Denmark, there's this dish called flæskesteg, that is a nice pork dish. Then the cook goes to the recipe book and knows, okay, it's a pork dish. I need a nice piece of meat. I might need a marinade. I might want to produce a certain sauce that comes with the dish. They describe how to do it. And then the cook will execute all the steps in the right order to produce this dish. The nice part about this cooking book, it's modular. Maybe there is a alternative dish that uses the same sauce. Then it's sufficient to describe how to make that sauce only once, to make a rule for that part only once. Coming now back to the genome analysis. In our case, I make a rule that says, please build a genome index. The input is our sequencing of the genome, the FNA file. The output, let's call it, is the index. And here is still a little bash involved to say, okay, make index for the input and the output, where these curly brackets indicate a placeholder, where the input and output that was specified will be pasted in. And on my shell, I type Snakemake, please make my index. Feel free to use all CPU cores that you have on your machine. Or I could limit it if I want to not have certain steps run in parallel. And it does not make a difference if I tell the name of the rule that I would like to invoke or the output file that I desire. Snakemake will determine then what commands to run. If we now transfer what we've learned about rules onto quality control, we have a raw data FASTQ file and an output file and, towards clean data that runs this quality control tool. We need an additional placeholder, a wildcard also, that is in the input and output file. So in this case here with a sample in the input is determined in the output. That is, if I write for Snakemake, please make for sample 1 the clean data. Snakemake goes through all its rule lists and uses pattern matching to determine which rule can produce the desired output. And notice if I set sample to sample 1, it can use indeed the QC cleaning rule that we produced that we wrote to produce the desired output. Going towards the more complex rule, for instance, such as the counting of expression, we can also name our input. So let's say I have one input that is the genome annotation and the second that contains a list of all the samples. Expand is comes from Snakemake. It's a way to expand for all possible entries in a list, which I provided here, the file. And I can refer in the shell script to these named input and parameters with the .genome. And maybe you're now familiar with, in Python, the formatting mini language. That is exactly what happens here. So these curly brackets are placeholders that Python, which is behind Snakemake, uses to fill the placeholders, either by naming specific entries, by referring to entries and maybe a specific position in the list. And if we, in case here in the bottom, only say input, it becomes a long list of entries that are just separated by a space. Now, that we have the part of how Snakemake might work, can we now have, please, an actual tool to work? Because currently QC tool is just a placeholder. It's not an actual tool. How would we be able to use it? And while I hope you appreciated this short introduction also with bash coding a bit, maybe it might be more efficient to just have a plug-and-play solution where we don't have to code as much ourselves. And yes, we have that. In Snakemake, there's the repository called Snakemake wrappers. And they have, for a lot of biological challenges, problems, a nice repository of tools for which I simply can copy-paste the rule, maybe adapt the input and output a little bit to match what I would like to have in my workflow. And thanks to this last entry wrapper, it indicates which version of this wrapper will be used, which when I run Snakemake with the use conda parameter will allow me to automatically install all the software. So maybe for the background information, what that means, Anaconda is a way how we can install in self-contained environments software, and we can install in different environments the same software in different versions, and we can use that also in a way to describe this environment to then go to a different machine, like a server, to install, recreate this environment there to run our code in a much more reproducible manner. And in this little example, this rule for this star tool, it's a mapper, or now an actual rather for transcriptomic data that we can use. And on the website, we also see the version of the individual tool of interest that would be used, which is of interest if we write an academic report. We can point out what versions of softwares we have been using. For our asthma-related transcriptomic dataset, I have on the GitHub tutorial a page where I've implemented a little workflow that takes for 4 samples, 548, 509, 512, and 513, the sequencing data, processes them with a QC cleaning, with a rule that I implemented, maps them against the genome for which we built the index, counts them, and combines everything in a single matrix. So if you would like to run through this little tutorial example from the GitHub page, you simply download it from GitHub, go into the subfolder and tell Snakemake, please build me the gene expression matrix, please parallelize steps that you can run parallel, use this conda mechanism to download the respective software, and also use Mamba as a front end to solve the software. For the context, if we use in this conda way of installing software and say I would like to install for this tool version 2.7, then it comes with certain requirements of dependencies, software dependencies that have to be met. And there is a so-called solver. It tries to find a solution which combinations of software to install to fulfill the environment that we desire. And Mamba is a fairly more modern, faster solver that was implemented such that if you run it on a consumer-grade notebook, it should run in its entirety a bit under 15 minutes. And the output will be a file now, gene expression matrix, which gives us the following entries per row, a gene identifier. So ENSG means ensemble gene. So there's a database called Ensemble where a lot of genes are being described, with this number being the specific entry in the database referring to the gene, where on the chromosome it's located, on which strand, how long entirely the genome is, and then finally for each of our 4 samples, a number indicating the number of reads that map to it. And since in this little tutorial I made a small subset of the entire data set, so it runs also on your notebook, there are only a few genes that are more highly expressed. For instance, this specific gene, if we look for it, then we see actual samples that have a couple thousand of reads. But that kind of is it to what I refer to as this pre-R analysis bottleneck. We have now a file, a symbol matrix with different columns per sample, per patient, and the genes that gives us the expression values. And now finally comes where I'm getting more excited for the analysis part now, to look into the data to see what is the biological signal that we might be able to see there. For this analysis, I'm going to use a lot of packages from the Bioconductor repository. If you've never heard of Bioconductor, it's a project. It's of a community of programmers, developers, and data scientists, biologists that would like to make a fairly open, welcoming community to produce software for high quality, reproducible, rigorous analysis of data in the biology and the life science. But even if maybe life science or biology is not the main interest of you, it might still be worthwhile to look into for the technical aspects of how they implement their infrastructure of packages. And even, for instance, Hadley Wickham in his Advanced R book refers to Bioconductor specifically if we would like to know how to effectively use object-oriented programming of the S4 system. So if we would like to know how that might work, Bioconductor is a great way to learn about it. In fact, in Bioconductor, there's also a package called Array, which gives us access to the full data set of this asthma-related transcriptomic study. We can install it with the Bioconductor manager and simply load the package and its related Array data object, which is now an S4 object of a ranged summarized experiment, which gives us a lot of the metadata, the counts, the gene names, everything in one concise format. So if we dive into it, there is a function called assay to access the assay, the test, the expression data from this array object. And if I look into, let's say, the subset of the first 5 rows and columns, it's this matrix of expression with rows being the genes and columns the patients or samples that I've mentioned. In case you're unfamiliar with these triangular symbols here, it's a short ligature of, if I type the vertical line and the greater than symbol, which gives in R these nice new pipes to make prettier code. So if instead of having to type g of f of x, I can say x, I pipe it into the function f, from which the output becomes into g. That allows us to make these nice flows of code across multiple lines. So I take the array object, extract the assay, convert it to a matrix, and then underscore referring to everything up to this point. On this matrix, I look at the first 5 rows and columns. Another function of interest is the col data function, which gives us access to the meta information for the column. That's why the name column, col data. And that is just a table which contains a lot of cryptic, more or less identifiers for different databases. We might recognize the SRR numbers that we used earlier to refer to samples. Here we see some additional connections that refer to specific entries in the larger database of the NCBI, of the National Center of Biological Information. For us, the 2 interesting columns are these 2, which tells us if this sample has been treated with a specific either of these 2 medications. Up in here, this line of code that produces the table that you see, I like to first convert the output from the column data to a tibble, which is a bit more of a tidy tabular format, which if we look at just the first 3 at the head of the table, I convert with the function cable in this nice figure that we see here from the knitr package. And in fact, in case you're curious, all the slides that you see are also made with R, specifically the R Markdown files. And for the more technical oriented R people, the little trick, this col data function is now actually the getter and setter for an attribute within the array object. If in case you've heard in S4 classes the slot, which in R we can address with this @ symbol, the call data of the array refers to what is stored within this slot of the object. Where there is a column data, there should also be a row data. And yes, we do have that. That gives us the different genes and their positions and their names. So if I have array, I would like to look at the row data, convert it to a tuple again. I will get a fairly large table of lots of columns. And if we just focus, if we select a subset of these columns to just look at the identifier name, the start and end position, I can then use, now comes the power of the tidyverse, use additional functions to, for instance, let's say I filter only, I would like only to remain with those genes that are on the X chromosome, have them nicely sorted by arranged by the starting position, and use from R the pretty number function to add the semicolon at 1,000 positions to have for me a more visually appealing display of the numbers. Now that we know what the overall structure of the array dataset is, now let's actually work with it. So one part on the left, let's say I use the 509 sample, focus on it because it was treated with this one drug, and make myself a little table that contains the gene identifier and just the expression within that specific sample. And I make myself a second table in which my genes are located, that is the name, the position, which chromosome it is on. Instead of cable, which produces this much nicer figure here on the left, in R we can often use the str function to look at the structure of an object. That tells us, okay, my genes, that is a tuple, which is this type of class. So in R programming, there are different levels of object-oriented programming. That is now an S3 object as a tabular data frame with the following columns, of which the last 3 have integers. Now of interest for me is to bring the information that we have in the left table and the right table together. And here we have a quite powerful tool, ally at hand, the join functions. That is, I have 2 tables and I would like to combine them together, to join them together on the gene identifier, which gives us one table that combines the gene names, the position, with the expression values. And those we can then use to make a little plot. So if I have my combined table of the genes and the expression data, and let's say I only look at the first chromosome and make a plot that has points for each gene. On the x-axis by position, where it starts, on the y-axis the expression, I get the following figure, which shows me a lot of each of these points being one gene along the chromosome 1. And I would immediately see, for instance, there is not a single point, there is no gene, no expression in the middle of the chromosome. In biology, chromosomes, they have these 2 chromatids structures, so we have the 2 copies of the same DNA together, combined with the centromeres here, they are glued together. Remember, DNA being the double helix of molecules, they are wrapped around, have a topology of the superhelix, get wrapped around against histones and become much, much more tighter, compressed, condensed, until we get this structure one could actually see in a microscope. And because they're very compressed and there's a lot of proteins that glue the 2 chromatids together in a chromosome, there is no gene that can be accessed and expressed here. And that's what we see in this nice figure, for instance. There is no expression here. Earlier where I said we would not just know where do we have expression, we want to see where is there a difference in expression. So in our data set, let's say I look at the 517 and the 513 samples. I make a so-called scatter plot that where each point is now again a gene representing and the expression levels relative to the two samples. And for instance, this gene here has a much higher expression in the 513 sample instead compared to the 517. So that's the first time we see some differences in patterns in expression. And if you might remember, I introduced you differences in expression are interesting because they can maybe be the reason why we see differences in how a cell behaves with respect to a drug, with a disease, environment, tissue. Let's take a step back and acknowledge we cannot compare these two libraries directly. Because here the blue line that I added is the diagonal, and we see how overall in 517 all the points are a little bit offset of it. So overall all the genes have a little bit higher expression in this library, so we can't directly compare them. How does that come about? Well, let's illustrate it in this example. I'm interested to know how many fish are there in a certain waterway. And I set out some people that go and collect samples and give me the report. I can't directly compare them. For instance, on the left, I have this gentleman with a small net, catches individual fishes out, diligently reported them, but the number is not easily comparable to, for instance, to the right, where I had multiple fishermen that had much larger nets capture in this much deeper pond lake fishes. So I can't compare these numbers. I have to make them comparable by a so-called normalization process. In RNA-seq data, what we can do is a size factor estimation. That is, I look per row, per gene on the geometric average. Each single entry, each cell in a row, I make the ratio to the respective geometric average. And then per column, per sample, I, on median, look into what is the ratio to the respective geometric mean. And this number, the size factor, happens to be a good estimation of how big, so to say, the net was which the sequencing machine used to capture the respective sample. In case you're interested to look up a bit more, I can refer to Anders and Huber's paper in Nature Proceeding, or this nice tutorial on Bioconductor that explains a bit more into the details of the steps. But to keep it short, now we have a single value. We can use that to normalize our data. And in fact, in Bioconductor, there is the ESEC2, differential expression sequencing package, that can do all these steps of normalizing the data and also make a statistical significance assessment for our model. So in our case, let's say I have the airway object, and there's now the beauty of Bioconductor, because it standardizes what type of data structure there is. This way of having a single file containing the experimental data, the meta information of genes, of samples. It can be directly plugged into a function from a different package to say, okay, I would like to make a regression analysis for the cell lines and the treatment with this DEX drugs, and run the normalization and regression and look at the results, which gives us then the differences as a so-called fold change. A fold change is if we look at the expression of the gene, let's say in a condition A, and the expression in a condition B, that could be healthy, diseased, or treated, not treated. That tells us the ratio of changes in expression. Let's say in this example, I have 3 genes. They all have 100 expression value after normalization in condition A. In condition B, the first gene has equal expression, and a twofold difference A versus B in comparison in the second case, and twice as high in the third case. And if we make the ratio fold change of A divided by B, we have the fold changes 1, 2, and 0.5. In our biological analysis, we prefer to use the logarithmic display, which tells us that 0, logarithm of 1 is always 0, which is representing no change. I think that's also mentally quite nice. If we have a value of 0, there has been no change. And we have a 1 in case of we use the logarithm to the base of 2, indicating a twofold change in direction, and plus and minus telling us then nicely, was it now in A or in B, twice as high. In now differential expression analyses, we are interested in these log fold changes, and we would like to have statistical assessment of significance that tells us how likely was it to see, given the data set, such a change by chance. How reliable can we identify differential expression, and also quite crucial to do what's called false discovery rate adjustment. Maybe you remember from statistics how that helps us with correcting for different errors. And all of that is implemented in this nice DESEC package. And when we take the results that we stored in this res object, I can make a quick plot of the log fold change against the log transformed p-value, which gives us this scatter plot, which is called a volcano plot. Why is it called volcano plot? Because it resembles a little bit this idea of we have a center of explosion and a lot of points going high up and into the far distance to the left and the right. And the further away from 0 a point is, a gene is, the higher the difference expression, and the higher up it's located, the more significant it is, something that might be of interest to look into. And in case of now coming to biology, that is what we do. We look at this data set, we make a call of differential expression, let's say at the significance cutoff of 0.05, the default, and select maybe genes that only had a log fold change of at least 1, so at least twice as high in either condition. Compare it together with the join to the meta information, and we can make a nice little list that we can then give to our biologist collaborators and say, okay, the gene number or gene names Spark L1 had much lower log fold change, so 2 to the power 4, higher expression change with a fairly low false discovery rate. And that is something where we can get excited and look into, okay, what do we know about this gene? Has that been described beforehand to be affected by this drug? Can that maybe give us a new insight into what is going on in the cell in the case of some sort of asthma-related disease? And that is where then the science really gets into it. I personally really like tables and CSV files, but when we work with biologists, often they prefer to look into an Excel file. So I could, of course, copy-paste the values from my R script into a file that I import to Excel, but luckily there's also the OpenExcelSX package that allows me to have a table that I can directly write into an Excel file that I can then give to a biologist. I can even add additional parameters that make different highlightings in a way that are of interest, because I, as a bioinformatician, I want to have preferably everything as a script, because I know once I've provided the report, automatically the reply will be, oh, what would happen if we use a different cutoff here? And I like to have it automized, so I just say change the parameter as desired, rerun all my scripts, get a report that I then can give to the shotgun wheeling biologist, who will be very, very happy. The last thing I've promised to talk about, how can we now, looping back to to the Snakemake workflow integrate an R script into it? Well, I simply, in my snake file, in my little cookbook, write, I have a rule for my analysis. It runs an R script, outputs, let's say, the volcano plot and my Excel file with the analysis results. The input was the data matrix. And as a little suggestion, the analysis script, the R script itself, should be also declared as an input. Because if I then go into the R file and maybe make a small adjustments, Snakemake internally notices, okay, this file has been changed. I now need to rerun it and recreate this, these files in order to be consistent with all the changes that occurred. And if you would like to go even further, if we declare or make a little conda environment description file, we can also explicitly list which R version, which packages, which package versions do we want to use for our script, and then add in our rule just a little declaration, conda environment, where we find it. That allows us then to also rerun this script on a cloud computer without having to worry about first installing the software and the right versions. If you're interested in knowing more about it, I can recommend you to look into the corresponding entry in the handbook from Snakemake. I would like to just point out there is no easy one-size-fits-all solution in expression analysis. I pointed out in the beginning when introducing this big data in life science approach, I've pointed out how our data sets might be very heterogeneous, and it depends a lot on what specific library design, experimental setup there might be. We then have to adapt our workflow or maybe create a new one when we want to handle all the analyses. I hope I was able to give you a good introduction into what sequencing data is, what it looks like, what transcriptomics is, why it's important to look into it, how in principle we could analyze it if we use the best script, but more importantly then, if we want to go bigger and want to have a workflow that allows us to scale to the data set size, want to make it in a way that becomes more reproducible and portable if we go to different platforms. And I hope I was able to give you introduction to the different packages that I personally use when analyzing transcriptomic data. And of course, here and there, some small tips and tricks in the R language. With that, I'd like to thank you for tuning in to this presentation. I hope you learned something, were enjoying it. And of course, I would like to thank my supervisors for enabling me to give this presentation. And of course, the Danish taxpayer for funding my work. And with that, have a great day.